Just as you might store birthday cards separately from bank statements, the brain keeps nice and nasty information in different places. A new study reveals pleasant experiences are stored in one part of the amygdala, an important memory center in the brain, while unpleasant ones are housed in another. The researchers say that the discovery of this filing system could lead to a cure for depression in the future. The research in mice showed positive and negative neurons in the amygdala are also genetically distinct, and they can even be manipulated to alter behavior. Scientists from the RIKE and MIT Center for Neural Circuit Genetics in Japan found nice experiences, tastes and smells are confined to the back of the basolateral nucleus (BLA), while unpleasant ones are stored at the front. These regions interact in a push-pull manner, regulating the behaviors tied to negative and positive memories. While the study was performed on mice, the researchers believe that it is likely the findings will apply to humans too. Dr. Susumu Tonegawa, who led the study, told, "Considering the well-documented evolutionary conservation of amygdala, it is very likely what we found in mice in this study will apply to humans. Previous studies have shown that a positive-negative division in the brains of mice and fruit flies for smell and taste memories, but this is the first time it's been suggested this split has been linked to behaviors to identify the negative and positive neurons." Researchers exposed male mice to an electric shock to the foot, a nasty experience, or the company of a female mouse, a nice experience. These experiences left their mark in the amygdala in the form of higher expression levels of the gene CFS, a marker of neural activity. The activated neurons were genetically profiled, revealing two distinct genetic markers, RPSO2 for negative neurons and PPP1R1B for positive neurons. The experts found that RSPO2 was observed almost exclusively in the front part of the BLA, while expression of the PPP1R1B gene was concentrated in the back of the BLA, showing that pleasant and unpleasant experiences are stored in different parts of the brain. The researchers also discovered the spatially distinct positive and negative neurons differed in size, shape and electrical properties as well. In the experiment, the positive neurons were activated when mice were exposed to pleasant smells and water rewards, while negative neurons became active in response to pain and unpleasant smells. The study, published in the journal Nature Neuroscience, revealed that positive and negative neurons are capable of disrupting a mouse's behavior if they are turned on or off. The rodents were trained to freeze on the spot when their feet were shocked, perform a nose poke when receiving water. The researchers found they could weaken these behaviors by targeting the positive or negate neurons with precise bursts of light, a process called optogenetics. Once the creatures were trained, they even discovered the different neurons could interfere with how well mice learned the associated positive and negative behaviors. For example, light stimulation of positive neurons during the foot shock training made mice freeze less, while the activation of negative neurons impaired the mice in the water reward task. This means stimulating the neurons could drown out the positive or negative meaning of nasty or nice experiences. Dr. Tonegawa added that at least for stress-induced depression, using these animal models could lead to a cure for depression.